Hello and welcome to the Encephalitis Podcast. My name is Prav, and I'm from Encephalitis International. Today, we'll be talking about the genetics of encephalitis. This is an important and new field of encephalitis research, and we are still learning so much. So, to get us up to speed, I am grateful to be joined by Dr. Sophie Binks. Dr. Binks is a clinical lecturer at the Nuffield Department of Clinical Neurosciences, University of Oxford, and an honorary neurology specialty registrar at Oxford University Hospitals, NHS Foundation Trust. Awarded a DPhil in autoimmune neurology, Dr. Binks is now a clinician scientist, and her research focuses on clinical and genetic aspects of autoimmune encephalitis. Dr. Binks was also Encephalitis International's first ever academic essay prize winner, which was the start of a long and fruitful and ongoing relationship. So I'm absolutely thrilled to be joined by Dr. Binks. Dr. Binks, welcome to the podcast. It's great to have you.
Well, thank you. And actually, I'm pleased that you essay prize. That was such a big moment for me. And recently I was sorting out my study and I came across actually some copies of the essay that I wrote back in 2011.
Wow.
And it was really kind of quite an emotional experience to go through those and think about that experience and the patient that inspired that essay as well.
Wow, that's amazing. Thank you so much. And I know you and I have also worked together. So yeah, it's just lovely to be working with you. So thank you so much. So this is a genetics of encephalitis podcast. So starting from the beginning, could you talk us through what genetics means and how it works?
Yeah, so most people will be familiar with the term gene and genetics and will realize that that implies DNA. And DNA is simply the body's code, as it were. So we have many different types of tissues and organs that make up our body, and we need to know a way that we produce those various aspects. And the DNA just tells the body how to do that essentially. So any protein that's in your body has a bit of this genetic code, i.e. DNA, that is responsible for the type of that protein that you have.
Great. And so, of course, the most important question I think will be on anyone's mind is we know encephalitis can be infectious or autoimmune, but can some people have a certain genetic predisposition or mutations that increase their vulnerability to these either of these types of encephalitis?
Yeah, so I mainly research autoimmune encephalitis, and we do know that there are some risk genes for some types of autoimmune encephalitis. But I'd like to stress, first of all, that this doesn't mean that if you carry some of these particular genes that you are definitely going to get autoimmune encephalitis. So for my clinical fellowship, I looked into something called human leukocyte antigen alleles. So this is a protein that we all have. Essentially, it plays a really important role in immunity and infection. And what we found was that about 90 to 95% of people that have a certain type of autoimmune encephalitis called LGI1 antibody encephalitis have a particular allele or variant of this HLA. Similarly, we found about 50% of people that have a different type of autoimmune encephalitis associated with something called contactin-associated protein 2 or Caspr2 have a completely different variant. So there's obviously a risk associated with carrying those particular HLAs. But actually, if you look at the population as a whole and thinking about LGI1, about 25% of healthy individuals carry this same HLA. And LGI1 antibody encephalitis is rare. We usually quote about 1 in a million. So clearly, having this HLA puts somebody at risk, but it is not enough by itself that you have to have this. And so therefore it's a risk factor, but there are very likely other things that come into play. These could be additional genetic variants, it could be environmental, for example, could be triggers from an infection in the past that can put someone at risk.
Right. So it sounds like it is kind of focused on this kind of HLA component. And I know that you recently published about how LGI1 antibody encephalitis has more than one of these genes associated with it. What are the sort of implications of understanding the sort of polygenic nature of this condition?
Yeah, so the initial research that we did was very much focused on HLA. But as I've said, there are likely to be other contributors as well. So a more recent study that I've done looked at a large group of people with LJ1 antibody encephalitis, and these patients were drawn from the UK, also from the USA and Ireland, and then there was a separate cohort of patients from France. And it's important actually at this point to mention that these sorts of projects are very much done as collaborations. There was a lot of people that worked on this project, including collaborators from all those countries. And this was something called a genome-wide association study. So this is a bit different. So in the HLA study, we very much were just looking at the HLA area. For the GWAS, you look at a number of, several million actually markers across the whole human genome. And what you're looking at is whether people that have a disease or a condition, in this case, LGI1 antibody encephalitis, have any of these markers that are more common at a statistically significant level compared to unaffected controls. So we had more than 200 people with LGI1 antibody encephalitis. And then we compared that to several thousand people from the UK Biobank who weren't affected. And we did find that there were some markers, and particularly there was one marker in a protein called PTPRD, which is enriched in the brain. And there was a certain variant that was more common in the patients than it was in the controls.
And when it comes to these genetic factors, do we also know about the relationship between these and more long-term complications after encephalitis? For example, seizures, memory problems, and personality changes.
So it's quite early days for that finding at the moment. We only released it earlier this year. And also, even though it was found to be statistically significantly enriched, it was found in about 5% this marker of the LGI1s compared to people without the condition. So we'd need quite a lot of work to unravel whether it is associated with any of those long-term complications that you mentioned. One association that we do know within the HLA sphere is that although there is this 90 to 95% linkage with this particular HLA called DRB10701 in LGI1 antibody encephalitis, there is another HLA which is called DRB1*0402. That is found, it's a rare HLA actually, and that's found a little bit in women with LGI1 antibody encephalitis. So we often think of LGI1 antibodies as being a predominantly male condition, about 2/3 men to 1/3 women affected in most cohorts. But actually, if you're a slightly younger woman, then this other risk HLA comes into play and is potentially linked to the condition in these kind of younger female patients.
Right, yeah. I was going to ask actually about the difference between male and female, and I think we see something similar with NMDA receptor encephalitis stuff, if I'm not wrong. So, what is the kind of relationship between all these types of autoimmune encephalititis at the genetic level?
So at the genetic level, the HLA predispositions are quite different. So as I've said, LGI1 and Caspr2 antibody encephalitis. In fact, quite interesting as relates to your point. At one point, they were actually felt to be part of a kind of similar or shared disease spectrum, and they used to be, and some listeners may have been diagnosed in this era actually, be collectively known as voltage-gated potassium channel complex antibody encephalitis. And for quite a while at the outset, they were thought to be kind of one and the same condition, really. We then kind of through laboratory experiments realized that they were separate. In most cases, a few patients actually carry antibodies to both LGI1 and Caspr2, but most are sort of either one or the other. We realized that this encephalitis was actually two separate conditions, and some patients had antibodies to LGI1 and some to Caspr2. And then it became clear with these genetic studies that actually the HLAs are also different. So as I've said, around 90 to 95% in most cohorts have this DRB10701, and in Caspr2 there's this different HLA called DRB11101. So it's quite a different risk factor, although HLA is involved in both cases. The other interesting thing, or I think it's interesting anyway, with caspr-2 is that this HLA-1101 may potentially only be found in those patients that have strong kind of central nervous system involvement. So caspr-2, again, some listeners may know, some patients present with slightly more what we call peripheral symptoms. So not as much affecting the brain, but you can have, for example, a painful peripheral neuropathy. You can have something called Morvan syndrome. Some patients get a cerebellar ataxia. There's quite a few different presentations, and some work done by the French group suggests that the 1101 kind of segregates with patients that have a strong brain involvement rather than those that have more peripheral nervous system symptoms. And to go back, I think we started off at NMDA receptor antibody encephalitis, which I don't want to ignore because it is the most common form of autoimmune encephalitis. So this actually predominantly affects young women, particularly of childbearing years, and can also affect children as well. It's quite a common autoimmune encephalitis in children. So there's not been such a strong HLA association found in NMDA receptor antibody encephalitis, but there are some other sort of genetic signals that are potentially coming through, and those are being looked at.
All right, yeah, looking forward to see where the research goes with there as well. And from the clinical side, are there any genetic tests that could help doctors predict encephalitis risk or guide treatment decisions?
So there's a few things that we can use in clinical practice, but they're not a kind of one-on-one relationship. So going back to kind of our original discussion about infectious and autoimmune, so some patients after HSV encephalitis can develop NMDA receptor antibody encephalitis. And there is actually an HLA, HLA A*02, I believe, which is slightly protective. So patients are a bit less likely to develop the post-infectious NMDA receptor antibody encephalitis if they carry this particular HLA. And in terms of the LGI1 and caspr-2 HLAs that I've talked about, so I wouldn't use those to make a diagnosis because they're both quite common in the population as a whole. So I said earlier, 25% of healthy individuals have DRB1 star A701, for example, and 25% of people in this country definitely do not have LGI1 antibody encephalitis. But where I think those can be particularly useful is when you're not sure. So sometimes, because no test is perfect, you might have a positive test result for either LGI1 or caspr-2 and not be quite sure if it's relevant to the patient's symptoms. We want to make sure that we're giving people the right treatment. Treatment regimes for autoimmune encephalitis are quite heavy immunotherapies, and that's great if that's what you need, but we don't want to put people that won't benefit from those treatments through those particular medications. So sometimes as part of a kind of a group of investigations, let's say, we can test for the HLA and see whether that then fits with various other factors. So not by itself, but maybe combined with things like imaging, lumbar puncture, and EEG, and then use that all together to come to a decision, actually, does this kind of overall picture fit an autoimmune syndrome that we know and that this person's going to benefit from immune therapy?
Yeah, so a holistic approach.
A holistic approach of which the HLA can be part of that.
Great. And we have a question from a member of our community, which is, does genetic predisposition to encephalitis influence likelihood of relapse?
I'm not aware of that. It could be an area of ongoing research. You could certainly think that having a certain HLA predisposition, predisposes you to develop these antibodies, and that therefore, if you kind of are in that situation, that it might make you kind of more likely. But I'm not aware. I will go and look up after this podcast. I am not aware of ever having used it in that context.
Yeah, that's great. The learning never stops, does it? And we're learning so much.
The question we sometimes get asked is it hereditary? And I actually want to sort of reassure on that point. So there's no evidence that it's hereditary. There are some very rare familial cases reported, but they're in the handful. So when I was writing up the manuscript on the GWAS from earlier this year, I think I came across 2 cases of familial LGI1 that had been reported ever. So, it's most things in medicine, you can never say never, but there's no kind of routine and common familial link.
Okay, thank you for that. And moving towards the end, actually, and one of the things that I've noticed in your research is that you have researched a form of spontaneously occurring autoimmune encephalitis in cats with the aim of improving outcomes in both species. So I was wondering what inspired you to undertake the study.
Well, I have to make a confession. I love cats. But I think it is a really fascinating, spontaneously arising animal model, actually. So these cats, people's pet cats living in the community, and they develop LGI1 antibodies and an encephalitis that has got very many clinical points of comparison with humans. So they have frequent seizures, behavior and personality change. And to my way of thinking, this is a really biologically important way of studying the condition across species. So, for example, I am actually looking at genetics in the pet cats because that will maybe provide extra information to understand why genetics are important in humans. It might also give an extra cohort, so to speak, to increase our power to discover causative genes. And I think that if we were to find some genes that were in common between the cats and the humans, this would give confidence that this was something that was really having an effect. And there was actually an example that some listeners may have come across because it was on the headline news quite recently, that a researcher in dog genetics found an obesity gene in dogs that was also relevant in humans. So that's the kind of way that I'm looking at it. And that I hope, as you so well put, Prav, that we can help both the humans and also the cats. So this is an animal model where the animal actually benefits from being enrolled in the research.
That's incredible. Thank you. And yeah, I love that kind of again holistic approach and yeah, we're also helping them as well. So,
Yeah.
it's yeah.
And it's actually a very enlightening working with veterinary neurologists. I think we can all learn from each other, and it just sort of widens our experiences and thoughts on the practice of neurology.
Absolutely. Yeah. And now I'm thinking of encephalitis as a multi-species thing. And yeah, it's just
Oh
broadened
well,
my horizon.
yeah.
Yeah.
Well, again, some listeners, viewers may remember Canuck the polar bear from Berlin Zoo that was diagnosed with NMDA receptor antibody encephalitis.
Wow. Yes. Yeah, so many examples. Wow. Yeah, thank you. And we have actually come to the end of this podcast, and I think we've talked about a range of things. We have talked about the heredity aspect, predisposition, different types of autoimmune, and even a different species. I was just wondering, as we come to the end of this podcast on genetics of encephalitis, is there anything that we haven't covered that you'd like to mention before closing?
So we talked a bit before we went on air, didn't we, about something that listeners may find interesting, that there is actually a genetic form of epilepsy associated with variants in the LGI1 protein itself. And we don't know, or we don't think that variants in the LGI1 protein itself are a strong contributor to LGI1 antibody encephalitis. But there's a little bit of overlap of some of the way that the seizures are in both groups. So, for example, in LGI1 genetic epilepsy, often people have seizures triggered by kind of noise or strong emotions. And we can see this in patients with LGI1 antibodies as well. So, I think there's kind of something there, a strand, and something that might kind of lead to some of the clinical impact of the LGI1 antibodies by kind of looking at this comparator group. But I just want to stress that they are distinct conditions, and we don't think that the autoimmune group is strongly influenced by genetic variants in the LGI1 protein itself. To our knowledge, of course, as you said earlier, we are always learning.
Absolutely, yeah. And I think it's a great way to end. I think there's so many different strands and so many different directions we can take research in, and hopefully we can inspire some folks to do that.
Yeah, very much so. I've got lots of things that I'm looking forward to doing. I will be doing sort of further research into autoimmune encephalitis, the genetics, and maybe look forward to catching up with you in another year or so with some new results, Prav.
Absolutely, yeah. Looking forward to see what we find out. Thank you. Um, yeah, so that does bring us to the end of our podcast. Thank you, Dr. Sophie Binks, for sharing your valuable expertise with us. It's been a pleasure having you on the Encephalitis podcast.
Oh yeah, absolutely delighted to be here. Thank you very much for the invitation.
And I would like to thank you, the audience, for watching and listening. If you have been affected by any of the issues raised, wherever you live in the world, please contact our support services. Encephalitis International is here to help in whatever way we can to achieve our aim of ending death and disability from encephalitis. For all our information, as well as details on how to donate, please see the podcast description or visit www.encephalitis.info/donate. Thank you again, and see you next time on the Encephalitis Podcast.